2 volunteers, and their names are Ashley, Divya. And Ashley is going to be your kinesin protein for day A, and Divya is going to be dynein. Okay, so what their task is going to be to move a purple, kind of spiky looking protein that is somewhere near the Golgi on my giant cell here. And they are going to insert this aquaporin into the plasma membrane. So first thing, by thinking about what things you needed to know, do you want kinesin or dynein to be involved in this movement, and why? So shout out, somebody have an answer? Dynein. Why do you want dynein? What do you... Okay, so you need to get it from the center of the cell to the outside. So what do you need to know about the motor protein? Does it go a plus end or a minus end motor? Plus end, exactly. It has to be a plus end motor because the minus end of your microtubules are near the middle. So we need a plus end motor, and that would be Ashley, who is Ashley, who is kinesin. She is your plus end motor. So now, can you help her find? She's a motor. She doesn't really know much. She doesn't have any eyes. How can she find her purple spiky protein? Where might it be? Does anybody know? Does a receptor help you? It doesn't. This protein has been made near the Golgi. Can anybody see where the Golgi is? Okay, it's over here. Okay. And do you remember what was inside this orange thing? Well, there were things that you actually expelled, and there were also some purple spiky things in there. So that actually is where the protein is that you need to transport. So she needs to transport that. Now, how, she doesn't have any eyes, how is she going to find that vesicle? What is it that the protein needs to be able to do? Receptor binding, yeah, it has to have a binding site. So you have to have a binding site on your hand to pick up your vesicle. Very good. And now she needs to get onto the microtubules. Where does she have to have another binding site? On her feet. Very good. So on her feet, she has a binding site that recognizes microtubules, which are those green things. And now you need to move toward the plus end because you're kinesin, and that is the only direction you can move. So she moves out to the plus end, and she has to get to the plasma membrane. And now she needs to insert her protein into the plasma membrane. It's in the vesicle. And so, yes, exactly. So it turns inside out, and now the protein is on the outside of the cell. So now we have our aquaporin protein out here. That's why it had to be inside the vesicle. It's made and packaged in the vesicle because it's inserted into the external membrane by turning it inside out. Okay, well now kinesin's done her job very well, but now we had 3,000 kinesins that put in vesicles and inserted this. What happened to our cell? It got big. It got really, really big, but our cells don't just continually grow forever and ever, right? So what other protein do we have to have? Dynein, right? So we need dynein to come over here. Dynein, a kinesin cannot go back toward the middle. She's stuck out here. Well, she might be able to get back up to the middle if somebody helps her. But dynein can take this vesicle back to the middle because dynein's good at walking on toward the minus end of the microtubule. Okay, gets all the way, and then it can be used in here it can be degraded, it can be used for other things. So dynein now did its job, but now we have all our dyneins here and all our kinesins there. So how can we solve this problem? Do you think you could carry her?
Maybe the opposite way around.
So what actually happens is that dyneins will transport kinesins along with other things back along the microtubule, and kinesin will transport dyneins back out. Other Otherwise, you'd end up with all your kinesins on the outside and all your dyneins on the inside. Oh, and one thing I forgot, they did all this movement, but they're very tired now. What else do they need? ATP. So they need energy to actually do their movements. So they get energy, and then they actually do physically move. These are proteins that do physical conformation changes, and I have you walking along here because, in fact, their motion is very much like walking. They have globular heads, and they're walking along the microtubules. Okay, thank you very much. Ashley,